Hi, today we're going to be discussing the relationship between TAM receptors and Zika-induced microcephaly. Presenting is Tammy, which is me, Harriet, and Crystal. And assisting us with this research is Dr. Ghosh. Some objectives for this research was to conduct a comprehensive literature review to understand the implications of Zika-induced microcephaly. To also look into mechanisms by which Zika crosses the placental barrier and depletes neural progenitor cells, also known as NPCs, leading to microcephaly. Third, identify potential Zika attachment factors, specifically TAM receptors, and test their role in the viral entry of NPCs. And lastly, to evaluate effectiveness of antibody blockades as a potential preventative measure against viral entry into NPCs. Overall, we want to contribute to the prevention of Zika-induced microcephaly through the development of monoclonal antibodies that neutralize the virus. Some key points to remember is that Zika virus depletes the NPCs within an infant, which leads to microcephaly. TAM receptors, tyro3, AXL, and MER, and their correlation to Zika virus, and the fact that there is no current treatment to help prevent microcephaly.
Zika correlation with microcephaly. Zika virus is a flavivirus transmitted by vectors, known for its link to microcephaly, a birth defect causing an abnormally small head size and brain development issues. Most cases of Zika virus-induced microcephaly were reported in Brazil during the 2015 to 2016 outbreak, with over 4,000 cases reported. Fetal infection during the first trimester is the main cause of Zika virus-induced microcephaly, as fetal neural progenitor cells, also known as NPCs, are vulnerable to infection. This virus can infect fetal NPCs by crossing the placental barrier, leading to cell death and impairing brain development. Ongoing research is necessary to understand how Zika virus enters host cells, as the virus remains a global public health threat despite efforts to control its spread. So why the interest in TAM receptors? TAM receptors, tyro3, AXL, and MER, are crucial transmembrane receptors involved in cellular functions such as phagocytosis and immune regulation. Recent research suggests that blocking Zika virus entry via TAM receptors could prevent Zika virus-associated defects. TAM receptors help viruses enter cells by activating signaling pathways and suppressing the immune responses. For example, they can suppress the production of antiviral cytokines such as interferons, thereby promoting viral replication and spread. More information is needed to understand the correlation between TAM receptors and Zika virus infection. This can help identify targets to prevent Zika virus and its effects, such as microcephaly. Targeting these receptors could provide a promising strategy for developing therapies against Zika virus.
Current studies indicated that the AXL viral receptor is in microglia during brain development. Since the AXL protein is most abundant within cells in the brain, researchers were able to inhibit Zika virus infection in glial cells by using MYD1, an AXL decoy receptor. Additional evidence supporting this was found in a study, which noted that enveloped viruses carry ligands that enable them to bind to Gas6 and protein S and engage with TAM receptors. As a result, they can attenuate type 1 IFN signaling, allowing them to evade host immunity. This indicates that the TAM neutralization could potentially be effective in combating flavivirus infections. Two studies have found opposing information regarding the TAM receptor. One indicated that there was a decrease in detection of viral genome for Zika patients.
Conclusions drawn from current studies. Although contradictory results exist between in vivo and in vitro experiments, the significance of TAM receptors in Zika cannot be easily dismissed. Rather, these findings illustrate the complex role of AXL in the flavivirus replication process. Further research should concentrate on identifying the reasons for the divergent outcomes between in vitro and in vitro experiments. By gaining a deeper understanding of these differences, the development of TAM inhibitors, such as monoclonal antibodies, could become an effective antiviral strategy for clinical treatment. Thank you for listening to our presentation.